All right, hello everybody. Welcome to the HIRM seminar. It's our pleasure today to have Dr. Jin Yu as our seminar speaker today. Jin received his BA in economics and his BSc in chemical engineering, amazing 2 degrees at the University of Illinois Urbana-Champaign back in the 90s. And then he went on to MIT to perform his PhD in computational neuroscience. He went after that to UCSD to do an MBA in entrepreneurship, which he's used very well in his career. And then from 2008 has been an assistant professor, then an associate professor, and now a full professor at UCSD. He is an expert on RNA processing and RNA binding factors and computational biology related to RNA biology. And so today he'll tell us about RNA binding proteins as regulators, drugs, and drug targets. So welcome, Jin. Welcome. The floor is yours.
Great. Thank you, Frank, for the kind introduction and the invite. I would have much rather be there in person, but as you all know, this is might take a while longer before we can do that. Let me change my background. I've been up since like 4 a.m. because I'm a lead organizing this RNA 2021 conference. And it's my fault for studying that on East Coast time. So let me go to something maybe a little bit less, something more relaxing. Let's see if I can do this. How's that? Okay, all right. So this is more relaxing, Maldives background. All right, so thank you for the invite, Frank. I'm pleased to discuss some of our recent work. And just before I start, let me see if I can get this going here. Okay, so quick disclosures of financial interest, labs funded by the Paul Allen Foundation, NIH, CZI, DOD, and Roche and Takeda. And so for more than a decade now, my lab's been very focused on understanding the roles of RNA binding proteins and how they control gene expression. There are, at this point, the numbers keep increasing. There are about 2,500 proteins that are annotated as RNA binding proteins encoded in the human genome. And as many of you know, they control essentially everything from birth to death of RNAs and how they're translated, how they're localized, how they're modulated even by a small non-coding RNA. So we're very interested in broadly using systems level approaches and very large scale approaches to understand what RBPs do. And over the last decade, we've been moving towards thinking about RBPs as regulators, drugs, and drug targets. And so I may present short stories here describing kind of the stuff that we've been doing in these 3 different areas. Okay. So RBPs in general exert their effects by interacting with cis regulatory elements. These can be short 4 to 6 nucleotide sequences. And by doing so, the RBPs modulate their RNA targets. And for the most part, functions of RBPs are unknown, and the RNA targets have been largely unexplored. And so we've been taking a very bottom-up approach in trying to understand the functions of human RNA binding proteins in a variety of different disease contexts and in development. And so we've leveraged a protocol known as cross-linking IP, or CLIP, first developed in Bob Darnell's lab in early 2000s. And we've been applying this with high throughput sequencing to look at targets and understand the rules of how our individual RNA binding proteins work to control RNA processing across different systems and a variety of different diseases. But in practice, this protocol is difficult and sometimes take several weeks with very high failure rates. And it's not always quantitative due to high PCR amplification. And so over the last, I guess now 8 years, we have been building up efforts to enhance this approach and also other approaches. And so just briefly go through Eric Van Nostren's lead on this in my group. It's now a faculty at Baylor, together with a grad student, Gabe Pratt, who's now in Seattle in a company and a large team. So the protocol that Eric and others had developed in the lab called Enhanced Clip, and it really features a really high diversity using significantly less material than previous approaches in the clip arena. And one good, very simple way of thinking about this is that if you download publicly available data sets and you just ask what is the percent of reads that are usable and not PCR duplicates, you know, on average is about 10–15%. So most of the reads are actually PCR duplicates in existing technologies in clip, whereas in our method here, using very enhanced ligations, you know, for each dot is a data set for RBP, right? A high fraction of our reads are actually usable, and so because of that, we're able to continue sequencing at depth and still maintain high diversity of newly discovered sites. We also feature input controls that are important to normalize the background, relying only on the IP data set here, where you look at RBP binding by clip isn't good enough. You have to always have a control as an experiment, obviously, right? So since our initial publication here, the lab has continued to make very small but useful changes for us, and finally culminating in a very robust protocol that enabled us to map more than 150 RNA binding proteins within like 18 months, right? In replicates and in 2 human cell lines. And I gotta say, this paper was just published last year, and we're late comer into the ENCODE consortium, because typically ENCODE focuses on DNA binding proteins and chromatin, and so this is the sort of first very large scale RNA binding protein effort, and already we have more page views than any of the other ENCODE paper, including the main paper in the same issue of Nature, and actually almost the same number of page views than all human ENCODE papers collectively combined. So pretty excited about the enthusiasm that the field is indicating that they're interested in RNA processing RBPs. And I just want to point out that this was a large collaboration with many groups. Chris Birch at MIT, I did my graduate work with Chris, did in vitro RBNS to identify RBP binding sites independent of context in the cell. And then obviously close collaboration with Brent Gravely's lab that depleted these RBPs to enable us to look at RNA splicing maps and the consequence of removing RBPs RNA processing. And then Shandong Fu doing chip for some of these RBPs, and as well as Lucas lab in Montreal that did a systematic screen on trying to evaluate what RBPs do at different subcellular localizations, right? And so it's a really large data set. I won't sort of spend too much time on this. It's really been published. Lots of groups are using the data set. Suffice to say that even a simple view of like, you know, where do RBPs bind in relative to like the genetic regions, right? Can tell us that some RBPs bind really to only the coding regions, right? Here, like the ribosomal subunits, which makes sense, RPS3 here, which will show up again later. And then some the splicing factors that bind typically in intronic regions, and then others binding to non-coding RNA. So very, very broad range of specificities. They don't, you know, they bind to, and by binding different regions, they control different steps of RNA processing. You know, a simple, also stark surprise for us is that some of these RBPs have very discrete and clear delineation points between binding upstream of the start codon or at the start codon, and then or downstream of the stop codon on mRNAs, right? And this, you know, enables us to actually identify rules of how they control mRNA stability and translation. Some, for example, bind near the start site, right? Transcription of start site. Others peak right at the start codon, and those are typically involved in translation control. and known to play regulatory roles like DDX, 3X, and so on. So the rules, a lot of these rules can be derived from these metagene profiles, and Eric recently published this in a accompanying paper to his Nature paper. The other thing that we've been very interested in is to derive protein-protein complexes and association from these maps. And so you can simply take binding sites for two RBPs in any of the two cell lines. We've been clipping these in and just comparing them and asking, do they correlate well? And by doing so, we can pick up, obviously, the U2AF 1 and 2. Binding sites are always next to one another, which makes sense because these are the 3' splice site recognition machinery. We can pick up known and new 5' splice site recognition components. We can also show that RBPs across different cell types actually correlate very well across different cell lines in this case, right? And then of course you can pick up some novel RBP interactions from knowing that they bind the same places. And we validated these and are moving into looking at large scale protein-protein interaction data sets now. So this resource has been, I think, useful for the community, I hope, because it's sort of the first systematic, same sort of protocol for what is the largest set of RNA binding proteins. And as we're pursuing this work, we were very interested also to continue to study other levels of RBPs, right? And so there are experimental, as I pointed out, deficiencies or difficulties with the CLIP protocol. Are there other ways to do this? My lab also looks at neurodevelopment, and we work at neurodegeneration, particularly in the ALS area. And we do a lot of organoid work and try to look at more complex systems. So can we develop ways to fill in blind spots where CLIP would require 20 million cells, or so high input requirements? And so when we're paying attention to other methods, we were very inspired by methods from Marv Wickens' group in yeast, where he fused RBPs to poly polymerase and uridylated additions, right, to RNA targets, enable his lab to identify targets for specific RBPs. And then work by Mike Rosbach's group in flies that fuse RBPs to the adenosine deaminase enzymes, enable adenosine to enosines, which can be read by SGs, right, in RNA-seq data, to inform us that these RBPs have been sitting at that site by using just RNA-seq, right? And so these are antibody-free methods. They don't require the same input amounts that CLIP does. There are pluses and minuses. And so we've been very intrigued about this, and we looked at some of the tribe data sets. And just to say that the tribe data sets were very sparse for us, not as sensitive as we thought. And I think that partly depends on the ADAR-mediated calling requires naturally occurring RNA hairpins. And so this is, was a sort of it may be a negative for us. And so we were thinking about different enzymes. We started fusing different ApoVec enzymes, and ApoVec edits any single-stranded RNA substrate not requiring double-stranded RNA or hairpins. And so as we were pursuing this, we saw Kate Maya's work showing that when she fused a small YTH domain to the ApoVec enzyme in the direct motif for M6A detection, she was able to detect these by seeing that the Cs are now used, right, and recognized in sequencing data. And so I guess great minds think alike. And so we were still very intrigued that it worked for this YTH subdomain. So what about full-length RBPs? Is this generalizable? Can we do something like that as we had done for CLIP, keeping something standardized and pushing it at scale? So one of my grad students drew this really cute cartoon here, just for us to sort of remind ourselves that these RBPs you can fuse ApoVecs to different parts of the protein. They convert Cs to Us, kind of like a parent sort of dragging a kid that is leaving trails here marking up where he has been. And of course, I think it's also important to have a control where the RBP is not fused. And so we call that the sort of the APOVEC only control. And so we tried this method with full-length RBPs, with new computational analysis here, which turns out required actually a lot of work on our part to enable really strong cluster identification of edits. And so we first evaluated this on RBFOX2, which is a well-described RNA binding protein. I've been studying this protein for more than a decade, right? So RBFOX2, as you can see from CLIP, eCLIP data sets binds in the 3' UTR of the APP gene. Input control is for here is clean. If you do control stem experiments, you can see that RNA-seq data sets are just in gray, and there's like maybe one edited site there if you induce high levels of APOVEC or higher levels, right? If you do this for RBFOX2, you can see that the edits do increase here, and if you just count not reads but edit percentages at particular nucleotides, you can see that that correlates reasonably well with where RBFOX2 binds, and we can derive different statistical thresholds and confidence to identify and clean up background from the control data set and still retain your RBFOX2 stem binding sites. So I'm going to quickly summarize some features, right? We think it's highly reproducible. You can apply stem to different replicates and show that you get very reproducible edits representing binding sites. We using our thresholds and a pipeline for identifying edit clusters, we call them. We can easily pick up the RBFOX2 motif, de novo, UG, CAUG. The edits are proximal to known CAUGs. I'm sorry about the pen, but it's like really peaky, whereas controls are flat. And it compares well with eCLIP data sets. So 50% of the stem data sets overlap with CLIP data sets, and vice versa is true. So it's not perfect. There are some plus and minuses the different technologies, but they're orthogonal. And so we can apply this to many RBPs. For example, stem loop binding protein binds the 3' ends of histone genes, and you can already see that the CLIP data sets here do require input to focus on to retain these binding sites at 3' UTR, but stem data sets are there. You can see the edits there, and then TIA1 is the same, right? You can see TIA1 edits, and you can pick up the de novo TIA1 motif, which is involved in stress granule biology. Now, one of the nice aspects about STAMP or antibody-free methods is that you can just subject this to different RNA sequencing technologies. And so one of the technologies we've been using a lot of in the lab is PacBio and Oxford Nanopore long read sequencing. And as an example here, this is the eCLIP data sets again, showing binding sites here on this track. And then the short read Illumina-based sequencing for RBFOX2 stem is over here. You can see that does overlap pretty well. And then in PacBio long reads, you can see that you also get a similar distribution of where the binding sites are. But the beauty of long reads is that you can identify isoform-sensitive binding sites, right? So with antibody methods, we fragment the RNA and leaving only the RBP protected fragment. Whereas here we don't. We can see that you have two different isoforms clearly delineated, and the long isoform is what's actually bound by RBFOX2. And this is the first time we've actually seen isoform sensitive binding of any RBPs. And I think this is a generally useful protocol. Now, what we really want to get into is to move into a single cell format, okay? And so you combine STEM with 10x, for example, because you can just do the RNA-seq on the 10x platform. And so these are the first views here of RBFOX2 in blue. These are the top 200 genes expressed in the 10x platform, and in blue is a percent edit in the bulk comparisons of like 5 to 20 million cells, right? 8% edits, and in green are the single cell aggregated edits that compare actually pretty well with the blue, right, in bulk. experiments. So again, bulk experiments, aggregated single cell experiments from 864 single cells. And this is sort of the first view of like where RBFOX2 binds to this transcript at the single cell level. Each track is one single cell. I mean, we were very excited because the first time we were actually seeing RBPs binding at single cell level. And then the aggregated view of this looks pretty good with even aggregated ECLIP data sets. And what's pretty cool is that you can get even to like one cell and identify the motif de novo from the binding sites of that one cell. So now we can sort of really do this for complex systems, right? Where you get single cell information on expression, but also binding sites. Now, perhaps not a surprise is that the ribosome is basically a collection of RNA binding proteins, right? And so we imagine that you can then fuse apobecs or different enzymes to ribosome subunits and then measure translation or ribosome occupancy. And so this is cribbing from our ENCODE work. We knew what our RPS subunits work well for CLIP. And so you can see the CLIP data sets here showing RPS3, as I mentioned before, RPS2 with DOX induction and binding sites in the exons for a coding gene. And for a non-coding gene, you see that it's clean. And keep in mind the ECLIP data sets still have noise here, right? That's why you have the input control to remove the non-coding background, but the STEM data sets are relatively clean here. Again, summary, you've got highly reproducible edits per km. So we call that, we sort of measure to do edits instead of RPKM, so edits per km. We correlate well with ribo-seq experiment in bulk and polysome-seq experiments. We see enrichment in coding, but also some enrichment in the UTR, I think from the apobec swinging out and the ribosomes piling up at the stop codon. But what's really cool is that you can treat with TARN1 inhibitors, right? And you can see that gene expression between vehicle treated and TARN treated is not different. But clearly there are genes which are very sensitive at the translational level to TARN1 treatment, which makes a lot of sense as a translation inhibitor. So I think it's sensitive to gene-specific changes at the ePKM edits per km level. And so, as I pointed out, you can combine what we call RiboStem now with 10x platforms to identify a individual single cells at the ePKM view, not RPKM. So you can see the RPS2 population there versus background. You can take the aggregated RPS ePKM measurements for the population and compares pretty well with polysome fraction data from 10, 20, 30 million cells, right? And so you can then multiplex also many different RVPs at the same time in a 10x experiment. So here's the first view of ribosome profiling, essentially in single cell level, right? This is RPS2 data. You've got some RBFOX2 targets and TO1 targets. And so we've been pretty excited by this. We show, and this just came online I think a couple of weeks ago. Here we show that STAMP can enable binding sets of full-length RBPs to be elucidated. Long read STAM can give you isoform-specific binding, and STAM allows really multiplex of RBP target interaction, site interactions, and allows detection of ribosome subunits at single cell level. And so again, Eric in my lab, talented grad students drawing this a cartoon of where this STAM sites are. What's exciting for us is we're already moving towards over the last year and a half now towards STAM 2.0 across a variety of different applications in mice and organoids and so on. And you can see that here's a cartoon that Hugo Medina, the postdoc in the lab, is showing that you can now use different enzymes as well, right? So this is the again MS2 coat protein enzyme fused to MS2 coat protein that recognizes MS2 RNA aptamers fused to an enzyme, and then we can do target RNA-seq to show that other edits around the MS2 stem loops, right? And so this is the CDU edits from STAMP. So you can see it's not it's good but not super strong in terms of signal. This is the ADAR hypertribe version, which is actually kind of blank. But then now we have other enzymes that do A to I, A to G, which is really, really strong signal, and we're curious about different applications on those. So that's just a quick overview of how we think about developing tools, I think, for studying RNA binding proteins at scale and systematically, but now moving towards single cell level resolution in complex systems and looking at, I will hope, multiple levels of RNA regulation. So moving on to next two little short talks is giving a sense of how my lab thinks about RBPs as drugs, right? And over the last couple of years, we had shown that RNA targeting Cas9 can work to recognize RNA in human living cells. We then developed, RNA targeting Cas9 to bind microsatellite RNA repeats, destroy these repeats, and showed that in both cells relevant to myotonic dystrophy, but also in mice experiments with AAV encapsulating the Cas9 protein and fused to an endonuclease to destroy RNA. And then obviously they're doing a bit more work in trying to build new editing enzymes with CRISPR proteins. But just suffice to say, these are just RNA targeting domains, and there are many RNA targeting domains that could be developed these days to recognize RNA specifically. The other aspect that we pay a lot of attention to is what are the effector domains that can be fused to RNA targeting domains, and I'll tell you a short story here. And the way we think about it is you fuse an RNA guided or protein guided RNA targeting domain module with a effector domain. Now you can develop RBPs as drugs, and we've been excited to push a lot of these concepts into a company that's in San Diego and pursuing RNA targeting RBPs as a potential drugs. Now, I mentioned that we are very interested in effector domains, and so again, I like to do things at scale systematically, and there's really not a lot of annotated functions for a given RBP. And keep in mind that our ENCODE efforts took quite a number of teams to do a maps for many proteins, but still many of these proteins are still unknown what they do. And so we made a RBP open reading frame MS2 coat protein fusion, as I've mentioned, library of about 900 over RNA binding proteins. And we set out to annotate what the RBP could do with the idea that domains within the RBP could confer a particular effect. In this case, we screen for stability and translation, but we screen for other effects on RNA targets as well. And so this is really taking a simple concept, tethered function assays, where you use, again, the MS2 coat protein tethered to the RBP and the domain, obviously the domains encoded in the full-length RBP to a reporter substrate. And this will have an effect on Firefly, mRNA, or luciferase protein output. And obviously we did everything in triplets here and then repeated the screen again, flipping the vanilla and Firefly as a control. So after many years of cloning and expressing cells and many plates, we were able to show that we can pick up targets. And for some reason, vanilla was much more sensitive, right, to Firefly, but we pick up many targets that what we call candidates that affect RNA stability or translation, right? The simple assay, we couldn't distinguish them, but then with QRT-PCR validation, we saw actually pretty good correlation between the RNA level of the target and luciferase, right? And so we picked out unbiasedly a lot of the known positive controls, right, that affected RNA stability or translation, and just list some of them here, including the C04, C07, deadenylation complexes. And then we validated a set of targets that when tethered to luciferase or vanilla flip RNA, reporter RNA, basically led to a reduction in protein and RNA levels. but we also did the same for ones we thought were very interesting, things that upregulated RNA and protein level. I mean, there are many ways to destroy stuff, but is there, what are the steps that enhance mRNA translation? So then we turned to our favorite tool. We did eClip on many of these candidates, and as with some positive controls as well. And just to show you that many of them exhibited this preference for binding the 3' UTRs of endogenous RNAs in the cell, which makes sense, right? Because that's how we sort of screen for them. But then they could control a stability or translation. Well, one of the things that one of the set of proteins that came up, which didn't do that, was this UBEP2L and MTDH protein. And UBEP2L, when we clip an endogenous targets, tend to be more in the coding region, right? And so we tethered it locally, but apparently in normal substrates, it's on the coding region. So what is it doing? This is the clip data sets on the gene showing control input and then the CDS. Okay, turns out that when we deplete UBEP2L with ssRNAs or CRISPR, it indicates that there are many RNAs that are stabilized, and also translation that was affected by UBEP2L. So it's a positive enhancer RNA and translation of endogenous genes, right? And we can use the sunset assay to show there's a reduction on UBEP2L leads to reduction in protein synthesis. This is using confirmed with UBEP2L knockouts in 293 T cells. Polysome profiling also indicated the same thing. In the knockout of UBEP2L, you see a reduction in polysome over input fraction by RNA-seq, right? Showing that UBEP2L is actually important for global translation. But also local, because you can tether, you can do what we've done with RNA targeting Cas approaches or 13 now to tether UBEP2L as the fusion to RNA that's probably never ever seen UBEP2L. This is like YP mRNA. And you can show that by doing that, you can enhance translation by bringing UBEP2L protein close. Mechanistically, we think that what's happening is that UBEP2L is binding to the ribosome and specifically to ribosome expansion segments. Basically, we think it's recruiting the UBEP2L protein to mRNAs, right? And so this is sort of showing that one of these expansion segments, ES31L, is highly enriched for UBEP2L binding, lies close to the region of the 60S subunit, which is occupied by tRNAs in the exit site during protein synthesis. And so this might be a potential way in which UBEP2L may be enabling enhanced translation. And so this was recently published, and this represents one of 3 efforts we're taking to decode. I mean, we only showed one protein endogenous levels, but we ended up with hundreds of different domains that can be fused to different RNA targeting proteins to alter the state of mRNA, as well as to understand what it normally do. And so the final part, again, just to showcase, we've been thinking a lot about RBPs also as drug targets. And part of my lab, as I mentioned, worked in the area of ALS. And so we've been very interested in looking for RNA binding proteins that we think are part of aggregation prone complexes that we figure if you identify these and you deplete them, you can enhance the survival of neurons that have disease-associated genetic mutations. For example, in ALS, Parkinson's, Huntington's disease, and so on. So one of our early efforts was to do apex proximity labeling in mass spec to identify proteins, particularly RNA binding proteins found in these RNA granules in cells. And then recently we've completed high throughput phenotypic small molecule screens to find compounds that targeted protein RNA aggregates and hence destroy granules and also enhance survival. Also recently a genetic approach using pooled CRISPR screens and imaging as a readout to identify genetic regulators, primarily RNA binding proteins, that will control the initiation and stability of RNA granules and aggregates in cells. But the other part of the lab is now moving towards focusing a lot on oncology. And we've been working closely with Frank Fornari and other investigators to do deep dives into the roles of RBPs in a variety of different cancers. And with the idea that we combine a lot of things that we normally do that we know, that we know how to do really well, which is to study what RBPs do, right? But in the context of looking for key vulnerabilities in oncology and in neurodegeneration. So a short story here from a grad student, Jackie Einstein, in my group. She's been very interested in triple negative breast cancer and where upregulated levels of MYC indicates a very poor prognosis in TNBC. And what has been described by MYC, right, is that these proto-oncogenic activating mutations often result in increased transcriptional and mRNA translational load. And so one would imagine that RNA processing plays a big key role here. And normally MYC is involved with controlling proliferation, but in a cancer cell, it also upregulates safeguard mechanisms in addition to transcriptional and mRNA translational overload that prevents the cells from dying from replicative and metabolic stress. And so one imagine is that if you can find these safeguard mechanisms, which we think of as really RNA binding proteins, you can create conditions where hyperactivated MYC cells and actually a variety of different cancer backgrounds could be subject to a therapeutic intervention. And so what Jackie did was do a CRISPR screen with a very focused library, really saturating RNA binding proteins, 10, 20 guides per gene for RBPs. And she did this in the context of a MYC tamoxifen sensitive line where you can treat with tamoxifen and translocate MYC to the nucleus, hyperactivating it and its function versus the control. And so she picked up obviously a lot of interesting proteins, interesting to us, and about 57 RBPs, of which I think 2/3 of them are actually highly upregulated in patient TNBC triple negative breast cancer samples, and then a third of them whose levels are lower in patients that survive longer with TNBC. And so one of these proteins she decided to focus on is the YTH-DF2 protein, which is an M6A reader for many reasons, including we have reagents in the lab. And so as many know now, right? So RNA methylation is M6A is a highly abundant modification in our mRNAs, which is there are writers and erasers to this modification, and there are also readers. And so YTH-DF2 is one of these readers whose levels are is actually higher in TNBC versus ERP or HER2 positive breast cancer. And when we deplete YTH-DF2, you get increase in cell death in the MYC-dependent cells. And just a couple of slides just to summarize this, right? We can divide cell lines into what the field recognizes MYC-dependent and MYC-independent cells. You can deplete YTH-DF2 and show that the tumors start slowing. Actually, we had to stop here because they became zero, essentially, after this time point. But then the MYC-independent cells were insensitive. What this meant was that we can do a xenograft experiment that dox-induce shRNA to knock down YTH-DF2 in vivo after tumors have already been formed and the tumors basically disappear. And what's exciting is that you can do I didn't point it out here, but you can do a full body knockout in adult of YTH-DF2 and mice survive. So not only is it specific, but also a safe approach to destroying the TNBC dependent, MYC dependent TNBC cells. We did a lot of work in looking for targets, including M6A sequencing and obviously ECLIP for YTHDF2. And we found that what's interesting was that there are a lot of targets, let's summarize, a lot of targets that are upregulated when YTHDF2 is depleted. Makes sense because it's binds the methylated RNAs and stabilizes them, right? But many of these were not actually YTHDF2 targets. In fact, in green are the actual CLIP targets, and there's only a smaller set, right? One of them is the strongest YTHDF2 direct target that's upregulated is PRSS23. So many of these were things which were secondary effects. So here's an example of what PRSS23 looks like. It's a serine protease, which is part of the intrinsic apoptotic pathway, and which is important for cellular stress, ER stress, and ROS, right? And so typically, PRSS23 is not present in this in cells, but in high MYC cells, YTHDF2 is upregulated, stabilizes this target, and so we find that when you deplete YTHDF2, you actually enhance intrinsic apoptotic pathway mechanisms. And perhaps what's kind of cool for us as RNA biologists is that we can do the single depletion. So when you deplete YTHDF2, you get increased cell death, so lower confluency of these cells. But when you do the YTHDF2 depletion and PRSS23 depletion, which is downstream, right, you rescue it. So basically, most of YTHDF2's function is through, we believe, PRSS23 mRNA stability regulation. So I hope I can convince you that YTHDF2 is a safe but also interesting target for TNBC, and we think it goes through this PRSS23 pathway. But I think there's also a few other targets that are interesting. But for now, what we think of is that this is a sensitizing one for TNBC. So we have since applied our approach to many different oncogenic backgrounds and are pursuing hard on the chemistry side to focus on developing ways to target these RBPs as therapeutic targets themselves. And so with that, I'm just going to close with the conclusion slide here. That I hope I convinced you that RBPs are an interesting class of proteins from the basic biology as drugs and as from engineering perspective and as drug targets. And of course, I want to thank my awesome team, the STEM team led by Chris Brennan, who's an amazing postdoc now on the job market. And of course, in collaboration also with Jason Underwood at PacBio. And then Hugo Medina is not on this initial paper, but is pushing on new advances in STAMP. And then on the MYC screen here, with collaborators Trey Westbrook, who provided expertise in vivo data on the modeling of the in vivo xenografts. And very grateful for everyone for your time and happy to take questions. And I'll stop.
Fantastic, Gene. That was great. Thank you very much. If there are any questions, please type them into the chat, or you can unmute yourself.
Or just ask.
Or just unmute yourselves and ask your questions.
Gene, that was a great talk. Quick question about the serine protease that's the target of the YTHDF2. Does that protease go back to affect MYC, or is that another more separated downstream event?
We don't know if it goes back to affect MYC. I think that's something that we're looking into, but we're evaluating that now. I think it's more general. I think is the idea is levels are really not found in normal cells, right? It's found highly upregulated in MYC dependent and actually other cancers, right? So it's a interesting one. Yeah.
Cool, thanks.
Other questions?
Yeah, Jean, there's a question from Jatayu Biswas. Hi, Jean. Great talk. Wanted to ask about the MS2 comparison of hypertribe and stem. MS2's stem loops are quite structured, double-stranded RNA stems. Any idea why ADAR doesn't seem to edit as well as APIP back in these cases?
Good question. I don't know. I mean, we are using, we don't know. I mean, it might be that the stem loops are occupied by the MS2 code protein that's binding them and sort of, maybe sort of preventing them from itself being edited. We don't know. That's data. Yeah, that's a good question. And APIP seems to edit it reasonably well. Yeah.
So Jin, from my side, okay, go ahead. There's a question from Gresham.
I have a question.
How's it going? Yeah,
I'm fine. Great talk as always. What do you think about the prospects of actually targeting the RNA binding proteins with small molecules? Because I mean, when I was talking with lots of people from pharma, they always discourage me. They say, well, this is undruggable, but what is your feeling? To what extent this is possible?
That's a good question. I think that there are three surprises for us. One is I think, at least for me, right, and you and others in the field have been working on this longer. We think that RBPs are actually interesting targets in the oncology and neurodegeneration realm, and I think that unbiased screens we've been doing actually point to that, and they're also safe. So folks always think that they're ubiquitous, they bind lots of stuff. So if you hit them, it might be bad for. No, I don't think that's the case. I think that they can be viable and safe targets in the adults, right? And then so that's like first two surprises, I guess. And then the third surprise, I think, is that these proteins are hard to drug because they bind RNAs with flat surfaces, right? I think that there are ways to do that. I think there are ways to identify ligands that can interact with binding pockets of these RBPs, and not all of them, but a subset of them. And I mean, I think that the problem there is that when you talk to pharma, they are colored by the history of all the RBPs that have been drugged by small molecules have been bad small molecules, right? HUR, Musashi, blah blah blah. But I think that, and I would agree with them. But I think what is missing is that a better menu, better options are RBPs. And I think if you look at the ones we've been looking at, I think those are absolutely druggable. So that's at least that's my hand wavy explanation there. Yeah.
Okay, thank you so much.
There's a question from Bruna. Bruna, do you want to unmute yourself?
Yes, hi, thank you. Very interesting talk. And I was just wondering about the APOBAC model. So do you see any difference in RNA conformation or translation or anything related to the RNA function by doing that addition? And the second question is it possible to use this model to trace any secreted RNA species, and have you done that?
Yes. Some good questions both. So does editing, you're right, does adding an APOBAC mess up the RNA? So in our current system, we don't induce them. We've made AAVs and lentees that can do that. We've also now ways to use small molecules to modulate the level. So we think that that number one, only a small fraction of targets are modified. So I don't think it affects the cell, right? Even for our ribosome subunits, right? It's only a small fraction, but it is quantitative and is distinct when you look at different conditions with treatment, for example, of cell types. So I think that it doesn't, at least in our hands, affect the survival or viability of the cell. So it does affect the RNA, but then it's an endpoint measurement for us, right? Then the second question is, can it be used to look at secreting? Yes, we have been working with groups to look into this. But if folks are interested, please come ping me, because I think that there are lots of interesting use cases here. Yeah, good. That's a good question. Yeah.
Question from Leon Wert Lamas. He says, "Hi Jane, very interesting talk. I would like to know if you have studied UBAP2, a very similar paralog to UBAP2L, and if you know what its function is.
Yeah, good question. So UBAP2L came up for us. Like there's like maybe 6 or 7 papers on UBAP2L to date, I think, right? And I think one of them is actually 2 of them from us now, and one of them we published in Sebastian's paper 218, where we characterized it as an interesting protein for RNA granule formation. We looked at UBAP. I don't think we looked at UBAP2 there, but now we're looking at both of them, and I think that they are likely to do something very similar, but we haven't completely characterized all their functions yet. Yeah, it's a good question. Yeah.
Thanks. A question from Ethan Dixon. Fantastic
Yeah.
talk, Gene. I'm curious, how efficient was your RNA knockdown using PIN-Dcas9 relative to the more conventional methods of RNA knockdown?
Yeah. So we in our initial paper, we evaluated PIN-Dcas9 on repetitive RNAs in the nucleus, right? And so these are expanded CUG, CAG repeats, G4C2 repeats that are expressed typically in the nucleus and cause myotonic dystrophy, ALS, Huntington's. Conventional siRNA knockdowns for us didn't work well for those repeats. And you know, that's a longer discussion, but we think that's because typically that happens, and Frank can tell us better, that happens in the cytoplasm. We also looked at ASOs, I believe, and ASOs for whatever reason didn't seem to work so well with specificity on the repeats for us. Whereas the Dcas9, we put a bunch of NLSs, it went in the nucleus, it bound these RNAs, and I think you know, the endonuclease, pin endonuclease is very effective. So I think in that comparison, it was a clear winner. Now in like individual mRNA, you know, gene knockdown, we would still, you know, for conventional mRNAs, we would probably still do conventional siRNA, shRNAs, or now Cas13d, or you know, not mediated depletions, right? So that's I guess in terms of tools, that's how we're thinking about it. Yeah.
Great. There's a question from Mariela Cortez Lopez. She says, great talk. I was wondering if it's possible to use STAMP to characterize modifications together with the binding using ONT's direct RNA-seq. And then she says, I guess in single cells it would be difficult, but I think in bulk it would work.
Yeah, I think the beauty of the STAMP system is that, again, for the first time, you can look at multiple RBPs or mods at the same time, right? So for example, you could obviously do, like we published also in RNA recently, like a nanopore approach to look at M6A sites, and obviously you can express in RBP fusion to look at where the M6A sites are and the binding on the same RNA on single molecule, right? So yes, for sure. But I think broadly you can look at 2 different proteins now on the same isoform, and you could never really do that in the past because you digest, right? You couldn't map it back, and I think you can do that now with this approach. And so we are busy, you know, putting together a lot of different new enzymes to enable us to do that. Yeah, good question.
Great. Please feel free to type your questions into the chat or unmute yourselves. Whilst people are thinking, I'll ask you a question. So Gene, have you taken advantage of the DepMap database to sort of prioritize RNA binding proteins that you might want to look at in cancer?
Yes, I absolutely. Yeah. So we have leveraged DepMap to look for which ones would be just, you know, bad to unsafe to deplete, for sure. Yes. We also have this, I mean, it's also important to realize that many of these RBPs have paralogs, right? And I think that may help us think about robustness in terms of toxicity, right? Where the dose is important, dosage reduction is important for a you know, particular oncogenic situation, but it's still not enough to cause toxicity to normal cells. And I think that's another advantage. RBP is a lot of parallels. Could be good and bad, right? Depending on how you think about it. Yeah. Good question. Yeah.
All right. Any other questions from the audience? Please unmute yourself. Okay. Well, if there are no more questions, let's thank our speaker, and thank you very much, Gene, for taking time out of your busy RNA symposium schedule, and it's
Thank
great to
you.
have your talk. And thanks to the audience for listening and for your questions. And we'll see you at our next event.
Thanks,
Bye,
Frank. Thanks,
everybody.
guys. Take care.
See you later,
Bye.
Jean. Bye, everyone.
See you.